Welcome to our lesson today. We are going to look at human eye. We have the diagram of a human eye. We have all the parts of the human eye here. We have the ciliary muscles, as you can see them, crystalline lens. This is the lens that we have in our eye. We have the cornea, the pupil. We have the muscles here. We have white of the eye. When you view your eye or your neighbor's eye, you will see the white part. So we have the part of the eye as stipulated below here. In the first part, this one we say that it is the hard layer that encloses the eye and is white. The white part that we see on our eyes or on our friend's eye is what we call sclerotic layer. Then we have the aqueous humor. This one is a clear fluid or clear liquid between the cornea and the lens, and this one, it helps to maintain the shape of the eye. We have the iris. This one is coloring of the eye. It has pupil, and this pupil regulates the amount of light entering the eye. We have crystalline lens. This is now where the refraction of rays takes place, and this one, it can change its focal length by the action of ciliary muscles when they relax or when they contract. We have vitreous humor. This one is a transparent jelly-like substance filling another chamber between the lens and the retina. We continue with the part. We have the retina. This is where the images are formed, and this is made of cells that are sensitive to the light. We have over there, this is the central part of the retina, and it exhibits the best details and the color vision at this place. We have the blind spot. This one contains cells that are not light sensitive. The ciliary muscles, this one actually supports the lens, and they are able to control the shape of the lens by either contracting or relaxing. In relaxing the muscles, it enables the lens to increase, hence it can be able to focus distant objects. So in other words, ciliary muscle will help to control the size of the focal length of that lens. In contracting the muscles, this reduces tension in the lens and it increases the focal length. Thus, objects, or we can say that near objects, are brought to focus, and this is what we call accommodation. You are able even to see near object because we have the ciliary muscles that are relaxing and contracting. Then we have another point. We have near point. This is the closest point which the normal eye can focus. A far point is the farthest point that a normal eye can focus. We move to the defects of eye. We have two types. One, we have short-sightedness. In this case, a person with this defect can only see clearly near objects. Rays from near objects are brought to focus on the retina, but those that are coming from a distant object, they are focused in front of the retina. The causes of this is that, one, you may have a short focal length, that is, the lens of your eyes has short focal length, or the eyeball is long. So we have this case in the diagram. We have that the near object are brought to focus correctly, but those distant objects, they are focused on in front of the retina. How do we correct this? We use a concave lens, or what we call a diverging lens, as you can be able to see here. So that now near object, the rays are diverged and they are able to bring into focus that image at the retina, so that we are able to view properly the objects that are far away from you. Then the second defect is what we call long-sightedness, and in this case, a person with this defect can see distant objects but not near ones. The images of the near objects are formed behind the retina as shown. So the causes of this is that you have too long focal length or too short eyeball. We correct it by using a converging lens like this, so that now the near object, the rays are converged and you are able to focus them on the retina. So that way you're able to correct that defect. From there we move to optical devices. We are going to look at three of them. We have simple microscope, compound microscope, and the camera. We start with the camera. A camera has lenses that focuses light from the object to form an image, and the image is formed on the film. Then focusing is done by adjusting the distance between the lens and the film, or sometimes you find somebody moving away from the object or closer to the object. That way he is trying to bring to focus that image. Then we have a diaphragm. This one is adjustable aperture, and this controls the amount of light entering the camera. We have the shutter, and this allows light to reach the film only for a precious period when you are taking a photo. Inside the camera is blackened, and this ensures that any stray light is absorbed. From there we can look at the similarities between the eye and the camera. One, we have seen that eye has a crystalline convex lens, while camera has a convex lens. Then the choroid of the eye is black. Then inside of the camera is painted black. We have said that this will ensure that the stray light are absorbed. Then the images on the eye are formed on the retina. Then in the camera we have the film. Then we have iris, controls the amount of light entering the eye, whereas in camera we have diaphragm, which controls the amount entering the camera. We look at differences. In the differences, we have that eye has a variable focal length. Remember, we have seen that the muscles, the ciliary muscles, are able to contract and relax. That way they are able to change the focal length. But in camera, the focal length is fixed. Then the image formed can change. We have the constant image distance. You can change you can't change the image distance for the eye, but for the camera it is variable. You can keep on changing. Then in eye you can constantly change the pictures. You can see several, you can move and get several pictures, but in case of a camera, only one photograph can be taken at a time. From there we move to the second device. We have simple microscope. This is called the magnifying glass, and it uses the idea where we found that when you put an object between the lens and the principal focus, that object is magnified. So in this diagram we have the object between the lens and the principal focus. You take a ray that is parallel to the principal axis, passes through F. A ray that is passing through the optical center goes undeviated. So if you extrapolate here, you'll find that the image that is formed is magnified. So when you use your lens in this way, then whatever you are having now is what we are calling a simple microscope, or what we call a magnifying glass. Then we have compound microscope. This one, it has two lenses. We have the lens that is close to the object, we call it objective lens, and the one that is close to the eye, we call it eyepiece lens. It uses these two cases. We know that we can only produce a magnified image when, one, the object is between F and twice F, and number two, when the object is between lens and F. So a compound microscope will combine the two, so that now you can get a magnified image. So we have seen that we have seen that it has two lenses. The focal length of the objective lens is short, and the one next to the eye, we have seen that it is called the eyepiece. The objective lens is of short focal length. So we have this kind of arrangement here. We have this is the object. Then close to the object we have the lens that we are calling the objective lens. If you look at where we have placed the object, it is between twice F and F. So the first magnification is done by the objective lens. A ray that is passing through the optical center goes undeviated, and a ray that is parallel to the principal axis will pass through F. So the first image from the objective lens is formed here. Then this image acts as an object to the second lens, The second lens, we are calling it eyepiece lens. So as you can see, we adjust the eyepiece lens such that we are able to get the object between F and lens. So that now we consider the ray that is parallel to the principal axis, that ray will pass through F, and a ray that is passing through the optical center, it goes undeviated. So if you look at the way they are emerging, these rays, you need to extrapolate on this side so that now where they intersect, you can be able to get your image, and this image is magnified. So this arrangement is what we call the compound microscope. And for us now to get the total magnification, we multiply the magnification of the first one and the magnification of the second one. So this is an improvement of simple. The simple was just using one lens, but this one now is combining the two lenses. So the total magnification is V over F minus 1. This is for the first one. That is the eyepiece. And this one is for the objective. So that way you are able to get a magnified object or a magnified image. Thank you, and let us meet in our next lesson.